Our first keynote speaker will be online. She joins us from US remotely. It's Professor Michal Rosen-Zvi, Director of AI for Healthcare and Life Sciences at IBM Research in Israel. She is internationally recognized for developing machine learning method for clinical decision support with a focus on real-world patient data and personalized treatment pathways. The title of her presentation, of her speech, is Beyond Words, Generative AI Meets the Complexity of Biology. So let's welcome Professor Michal Rosen-Zvi. We'll see her soon.
Thank you very much. I'm happy to join. Do you hear me well?
We hear you here in Prague.
Perfect. I'm very, very happy to be here. Thank you very much. Thank you for the invitation. And I would like to share with you today some highlights from the generative AI technologies that have been developed recently and seems to be highly valuable for research of brain and research more generally of biomedical data. And to start, I'd like to go back in history and to highlight the importance of algorithmic developments in that are based on mathematical breakthroughs. I don't know how many of you remember the breakthrough from 1964 of the Cooley-Tukey fast Fourier transform algorithm. This started with a very interesting story about dark times of Cold War between US and Russia, and the question of US President Committee that wanted to track the nuclear weapons testing done by Russia. And these were relying on huge sensor data, and the challenge that the committee put in front of some researchers was, how can you analyze this data very quickly and accurately? And that's how innovation came through this pressure on finding the answers to these challenges. As we all know, and definitely in CLARA, you have a lot of activities. Today's technologies are not only using classical data, classical machines and generative AI technologies, but also quantum technologies. And what I want to share today is how this evolvement of technologies of quantum and AI can be driven by needs from the biomedical world, by the pressing needs of resolving conditions and diseases like the ones every one of you is tackling every day, but also that there is a need to map out the data complexities into mathematical framing that will enable algorithmic innovation and usage of these algorithms to accelerate the discoveries that are needed. So this is the key message, but I will take you through different parts of it. Some of them are more generic and some of them are very concrete. So I'll start with the broader view. If we think about the mathematics and the challenges we've been learning when we were doing I did PhD in physics, but whatever science or STEM you've learned, you've probably, and I know you have a delegation of students, you probably tackled it in your everyday, but most of us, it's in the past. So when we think about the challenges, how science is being captured with mathematical equations, very broadly, there are 4 different ways. There are differential equations that capture dynamical systems. Everything we do in the world are dynamic systems, and differential equations capture that. Linear algebra and eigenvalue problems capture a lot of what happens today, definitely when you think about Schrödinger equations and quantum way to represent what happens in the world, as well as Newtonian equations. And then there are often the challenges in the world are captured through optimization and combinatorial problems. And last but not least, when we analyze data, often we model that as some noise that comes out of, some noise added to a signal, and we ask a question of probability and distribution, and can we estimate the probability distribution, and we assume some statistics on the data, and that's the way we capture the information. So these are like the very broad ways of capturing capturing data, learning from that through mathematical framing. And Feynman, in his Nobel Prize lecture in 1965, talked about how when you try to resolve challenging questions, it's better to have different framing thinking about these questions. So paraphrasing what he was saying in his lecture, we can think that I believe that a good biomedical researcher might benefit from having multiple mathematical perspectives on the same problem, as this diversity can inspire breakthroughs, algorithmic approaches that accelerate discovery. And if I got it well from all these nice opening comments, we are all in the business of trying to drive discovery to address challenging questions in the context of neurodegenerative conditions and better health for populations. And when we think about the advancement that happened recently, now I'm living the history, the past history, and coming back to today, in the past decade, we have seen amazing breakthroughs in the ability of classical AI technologies to comprehend language and to analyze language and to analyze natural images. And in fact, many benchmarks and tests have shown that these technologies are at the same level of human capability in writing, answering questions, and so on, and even when the questions are in the mathematical domain, but in a very, I'll call it simplistic or school level of mathematics. The big challenge that we have today is to resolve the questions of mathematics, physics, chemistry, optimization, algorithms in the world of the science, in the challenges that we are tackling when we are learning from data and try to make discovery. And the expectation is that the innovation will come with new algorithms. And it doesn't matter if you think about a quantum computer or a classic computer, the algorithm on top of the computer, the software that needs to evolve and to address the challenges, very generally can be divided into how you represent your data and what abstraction you apply to this data. How you represent the data, these are the typical questions. If you have, let's say you are looking at an image, MRI image of the brain. So you might look at the pixel level, you might have a different representation, even you might apply AI technology that condense the information, and then you have representation from the hidden layer in your deep learning algorithm. So that would be the presentation. But then there's the abstraction. What do you assume about the data and how you frame the challenge that you tackle? So it might be, let's say if it's MRI, you want to tackle signals of early Alzheimer. And you might have an hypothesis about accumulation of certain proteins. So the framing, how you abstract the question, are you looking to optimize detection of certain areas in the image? Or are you looking for the typical distribution of these proteins and deviation from this typical distribution? So there are different abstractions that one might apply when they want to create an algorithm that will provide an answer, will enable discovery. And when we think about molecules that we learn from, whether it's proteins or small molecules, and I'm now taking you directly to the world I'm part of, which is the biomedical discovery world, when you think about representing data coming from molecular information, there are many ways to represent that. You might have free text that describe what protein you have in mind, or what DNA, or what ligand. Or you might have a sequence of the basic components, whether it's the amino acids or the or the DNA base pairs, or is it the representation of selfie or smiles of the ligand? So you take the molecule and break it to its atoms and use some standard way to represent it with a sequence. Or you might have a 2D capturing of the molecule, a 3D, or maybe even capturing its dynamic, and that would be a 4D representation. So many of our studies that focus on biomedical data would be at that level. We want to analyze proteins or therapeutic. Obviously, when we move to the cell, there are other ways to capture the cell, but it's the same concept, whether we want to look at the expression levels of proteins and we just use counts, or we use some other ways to represent and capture the information about which protein or which messenger RNA copies are very abundant and which are very come in small numbers in a certain data. You can again use free text to represent that or any other ways. We can think about many of the classical methods today as a way to compress the data, to take the original large-scale data and whether it's through rational autoencoders or any other method, you compress the data and now you have hidden layers that provide you a representation. But at the end, after you took all of this complex data and you represented it, then comes the challenge of framing it using some abstraction so that you can resolve the challenge. And I give here some examples, but since I will come back to them later, I would not elaborate now. I just want to make a point, because I know some of you are working with quantum computers, some quantum computing, and some with classical methods, that this point of view shared by Jay Gambata from IBM Research, who is who's the director of research, but also leading the quantum activities, and by Alessandro Corioni, who's leading the applications, solutions through quantum and AI. So this view holds well for both ways of addressing algorithmic development. It doesn't matter if at the end you are using classical machine or quantum machine, phrasing the challenges through this prism of mathematical way of what framing you choose to focus on enables development of a wide range of algorithms that fit these different framings. In the biomedical world at IBM Research, we've been working on developing foundation models that capture this data and its various representations, and we've been doing that in collaboration with many different partners. And as I heard from the director of it now in his opening, collaboration is very important for us, and we've been working with many partners, and I'm very happy for potential collaboration here as well with INDRC and CLARA. And many of the technologies we've developed are open sourced and shared externally, both in GitHub and in Hugging Face. So you can download the models and you can also use the code. So I'm going to very quickly share with you 4 different models that we've developed. Each addresses different aspects of potential biomedical data and enables different ways of learning from the data. From the representation point of view, we've been looking at sequence representation, image representation, 2D graph and 3D structures. What I'm sharing now is what's already out there. We are making progress on these fronts also these days because, as you can see here, the way we represented the data to learn across DNA, RNA, protein, and small molecules, the easiest was to capture all of that through sequence representation, and that enabled a nice development. But then we have expanded that to learn about DNA and RNA, and we moved away from only sequence representation and captured also the numerical values in various ways. What we've been doing in order to make discovery is to first use the paradigm of foundation models. I trust you all aware of that. It was The term was coined in 2020 in a Stanford interdisciplinary group that was established to identify what's new in algorithmic development in deep learning and so on. And at that days, they captured foundation models as a breakthrough that has certain elements into it. One aspect of it is that it's based on deep neural networks, the innovation that came forward with Jeff Hinton, the Nobel Prize laureate of last year, who showed that backpropagation is an efficient way to learn from neural networks. So, and many others in the community. So, deep learning is a strong algorithmic way to analyze data, and then they have shown that with certain progress that was made by many different groups, that if you pre-train and use masking approach, so you take huge amount of data and mask some of the data and learn to predict the masked data, so this pre-train approach is very effective and attention mechanism is very effective. So using an architecture that captures certain areas and gives them higher weight in a certain method. Generally, that's what they called back then foundation model. These days, there are many other algorithms like diffusion models and others who are being deployed by the community and has certain similar elements. And in the world of biomedical data, where you want to make innovation and resolve questions associated with health and disease, there are a lot of open databases at the scale of millions and billions of data points. And what you see here are all open data sets that we've been leveraging in our studies. Everybody can use them. And I divided them by whether they capture small molecules, proteins, gene expression, or DNA. Small molecules, there are billions of data points. We have been using a subset of that we've cleaned, that we identified as relevant for drugs, because small molecules can be anything, can be used in many, many different ways. Proteins, obviously, there are many different databases, whether antibodies databases or protein-protein interaction databases, and you can see here a list of databases that we've been using. And similarly, recently, gene expression data was made available at CellXGene and Pangolo and many other resources, and there's these days more data sets at the level of hundreds of millions that we've been using that were recently shared by Taho and another team. And the DNA data, we use the human reference data and DBSNP, which provides you information about data. So all of these data sets were leveraged in these 4 models that I mentioned that I would share with you. And you can see here the 4 approaches. You can find out more information about these papers that are all under review in different journals. And also, you can find them, as I said, at Hugging Face and so on. So what I'll do in the rest of the time, I'll take 1 minute break, because I heard that you are highly encouraged to ask questions. So I'll take 1 minute break if someone wants to intervene now. And then I'll take you through what we've been able to achieve with these models and how we see the future with agentic systems. So 1 minute break. Any quick question?
Any questions? Looks like not for the moment. No questions so far.
Thank you. So I'll go forward with the 4 models. The first model, as I shared before, is learning from different views and suggesting how we can estimate interactions between the small molecule drugs and their target proteins. So there are many different methods to evaluate such models based on, can we predict their properties like toxicity, and can we predict their properties like brain-blood barrier penetration, and many other benchmarks that the community have created. There are challenges with these benchmarks, and we've been working with different teams on discussing and expanding the benchmarks so that they capture better reality. But like the rest of the community, we did tested the models through the benchmarks and showed that our approach of combining different views of the molecules does provide benefit. And we are now working with Professor Fei-Shang Cheng from Cleveland Clinic, who is expert in Alzheimer's disease and interested in finding small molecules that can potentially be beneficial for Alzheimer's. And we are looking at existing small molecules and asking whether any of the small molecules can be repurposed or can be leveraged, if it's not yet used in practice, against Alzheimer's relevant targets. So this is a study that we are now performing in the lab. In the second model, mammal, that looks across different modalities and learns from many modalities, we can answer questions of antibody infilling, antibody-antigen binding, protein-protein interaction, same as I said before, the brain-blood barrier penetration, toxicity, and so on, because now we have very broad view on many different domains. We specifically advanced this model to be very strong for protein design and for protein properties prediction. We tested that even against AlphaFold 3. You can see the results in our paper, and we showed that for certain targets, the ability of AlphaFold 3, as we know, AlphaFold is a very strong technology that was introduced by DeepMind 7 years ago and won in competition in their ability to predict structure of proteins. And what we used with AlphaFold 3, which is used by the community, is to estimate stability of a complex. So the question of binding of proteins might be mapped into whether these two proteins create a stable complex of proteins. Are they bind together and stay together? And this is being used by the community using some values that the AlphaFold 3 offers. And we tested that with colleagues from the Technion University and showed that our mammal algorithm that is shared externally and everybody can use it outperforms binding estimates of certain antibody-antigen complexes. So outperforms AlphaFold as well, AlphaFold 3 as well. In the RNA world, the challenge is the challenge that many teams have been applying is the challenge of characterizing a cell through the profile of gene expression. So if you have gene expression, single cell RNA-seq data from a cell, and you want to know what cell is that? Is this a brain cell or maybe brains, typically is the nucleotide, but is it a liver cell or a lung cell and so on? And you want to look at the profile of the gene expression and to identify the source of the cell. That's a typical task that is well handled by many algorithms recently developed in the past 4 years for single-cell RNA-seq data. Same approach of foundation models, same approach of pre-training on millions of data. And what we've done at IBM is we have been looking at all the advances made by other groups like SCGPT, which is a very strong algorithm, and we have developed a framework that enables using many different ways of representing the data and analyzing that. We are using the framework, so as we said before, there are different ways to represent data, and what you see at left at the upper panel, number 1, are different ways to map the RNA transcriptomic data into different representations. Then there are different ways to capture the data. You might think about it to frame it as a probabilistic question, and that's what we've been doing, and capture the numerical values and ask about the noise. And we've been showing that our methods, comparing to other methods, well distinguish between people with certain conditions. So people, control case with no condition and multiple sclerosis, the gene expression profile data analyzed with our methods the algorithm can distinguish between healthy and people with this condition. Similarly, we've been suggesting a novel way to represent DNA data and the SNPs, and we are analyzing and benchmarked against different methods. And what I want to share with you now are specific applications. Let me just check the time. How much time do we have? I have like 5 minutes to complete.
Just a very, very little bit, but 5 minutes is very okay.
Thank you very much. So what I'd like to share with you are certain studies that we've been doing with these models and how we were able to accelerate discovery with partners using these models. One question that was that we were challenged with, that we were tasked to analyze, is a question of how good an organoid is in capturing the reality of patient's organ. So the organ we focused on was the gut, and the question was about IBD, about severe IBD. And a colleague of ours, Professor Tad Steppenbach, is putting this question in place because he's developing organoids to enable studying the condition. And the organoids are created from certain cells that are being, certain set of cells that keep changing their flavor and nature, and you want to ask whether the different cells capture what you expect to see in the gut. So you want to analyze the transcriptomic data coming from the organoid and compare that to transcriptomic data coming from real patients and ask about the distribution of the different cells and make a comparison between what you see in your organoid to what you see in the patient data. And what we've seen is that our, what we call BMFM RNA, is a very useful tool to compare and contrast between the organoid and the patient data. And this is now under review in some journal, and you can find the paper online. Similarly, we've been working with Professor Fishong Cheng that I mentioned previously, and we use our methods in order to provide candidate for repurposing for Alzheimer's conditions. The screen compound is what I mentioned with Melon. It's the questions about the small molecules. And for the mammal model that I mentioned before, we've been using that for vaccine design with partners who are looking for binding affinity as part of the questions of whether they have a good antibody candidate for influenza vaccine. And now I'm wrapping up. So I gave you some examples of the models and how we apply them, and I want to wrap up with a key aspect of what we've been doing. So as you know, foundation models, these huge models that learn from data, are sort of a black box. They are not self-explanatory. It's hard to know what exactly they've been doing. And that's why we massively benchmark them, because that would be a way to make transparent their limitations and capabilities. And this creates a strong dependency on the benchmarks. We are looking for other ways to explain and understand the models so that you can create trust and benefit from these models. And very high level, when you think about what are ways to create explainability in AI models, there are 3 different methods. There are models that are inherently explainable. For instance, decision tree. If you want to know what a decision tree did, you can just go through the different leaves in the tree and you understand the process. So that's easy for human to grasp. Many of the models are not like that, and to understand them, you do a post-analysis. In deep learning for images, like we talked about the MRI before, if you ask questions about what you see in MRI, and you want to know how the model worked, some methods are using masking. So if you hide some of the model and you see if the answer changes, it gives you an indication of which pixels play the most significant role for your answer. So that's a post-analysis method. And there are many different post-analysis methods. I trust you're all familiar with Lime and SHAP, and there are many others like that. And the last method that is rarely being used is causal analysis, where you try to see what caused an outcome and pinpoint this cause with algorithmic approaches. These are very challenging methods that people are developing. What we are relying on when we think about, okay, let's just say I'll skip this one because I want to finish. You know what, I'll just So what we are relying on when we want to analyze our capability to predict with models and the strengths of the models are two strong elements. One are benchmarks, and the other one is trying to offer explainability through literature and other resources, and that's being done with agentic systems. So for benchmarks and transparency, we have a lot of efforts in the open with collaborators. We are participating in part of the Structural Genomic Consortium, and I'm highlighting here a challenge, a dream challenge that IBM is involved in, as well as many other partners, that is around benchmarking capabilities of models of understanding small molecules binding. We are also part of an AI alliance, which is open to the full community. Everybody is welcome to join us. We have had 2 events in October 2024, in September 25, and this is a community, the AI alliance, that includes more than 150 different partners, and we have established an AI for drug discovery working group where everybody is invited to join. And agentic systems, we've been working on methods to connect through agents. So the models I showed before, some of them are wrapped with MCP, which is a way to apply them remotely. And I won't explain what agentic system here in this opportunity, but I'll just mention that if you want to automate the usage of the models and to couple them with some explanation from the literature, you can do that not only leveraging existing LLMs, but also using our models and link between the two in order to achieve some transparency and understanding the technology. And with that, I'm finishing. Thank you very much for the opportunity to talk here today.
Well, we thank you. Thank you for a very interesting, deep, and important battle in the whole story. I'll just ask if we have some questions here, and looks like we do have some, so.
Yeah, thank you very much. I don't have question. Actually, I would like to thank you, Professor, for this lovely keynote speech, and on behalf of INDRC Director and also CLARA Supervisory Board Chair with Jochkal, who is currently giving an interview to Czech radio, so he wasn't able to thank you very much on himself. So this was just from my side. Thank you very much.
Yeah.
Thank you, my pleasure.
And we have some questions here from Slido, so let me read it. Let's choose this one. I love that we are integrating research from neuron to nation. How can we intentionally engage patients, their families, their care providers, and payers? That was the question. The full one.
I'm happy to share my view, but this is a very ambitious challenge that I trust a lot of the experts in the room have their own viewpoint on that. So the challenge of bridging the gap between research and at the bench side, at the translational, this huge gap of the last mile is very daunting, and I know that we all the researchers are potentially patients, and we all have families, and we all want to see that what we've been doing goes into the hands of patients. That's definitely a huge challenge for us. In my view, the way to address that is exactly the points I was making of being transparent, open, sharing the model, showing the code, and working with communities like INDRC and with like the AI Alliance, and making sure that everything can be captured and tested well. I'm optimistic that with the advances in AI, and hopefully also with quantum to address some of the most challenging parts, but definitely with the AI and the agentic system that I just touched upon very lightly, there would be a faster way to consume and validate the technology. And it's a challenge we are all part of, and as I said, I trust people in the room have much more experience on bridging the gap. I'm doing my efforts from my side, but I'm really remote from the payer and patients in that sense, because as a research organization, we are at the research part.
All right, thank you so much. Looks like there is a lot of questions. There will be lots more in Slido. I can see them already. But let's give first one we have here.
The title of the meeting is Unlocking Complexity. And one of the challenges for neurodegenerative disorders is to unlock the rules by which emergent behavior occurs. For example, cognitive impairment is an emergent characteristic of systems failure. The question and the key challenge is, how do we predict what's going to happen at the upper end of the complexity by the knowledge, tremendous amount of detailed knowledge at the lower level, at the granular level. For example, enormous knowledge about the primary structure of a protein or its secondary structure. In what way can we use that knowledge to predict its folding characteristics? What are the rules by which changes in the amino sequence leads to what structural topology, how the topology of the complex surface of the folded protein emerges? What strategy should we be using to understand the rules by which we go from one level to the next level? Can you give us your thoughts on that? That would be the kind of thing that I think CLARA would want to do with you and your group's collaboration.
What you described is a real challenge that we are looking at, the emergent behavior and the ability to predict these, I would call it maybe a phase transition, because as you said, the emergent behavior changes completely the way the brain behaves. And when we talked about the mathematical framing, we think about it from different angles, and we are looking into ways to characterize ahead of time the system. And as we know, there are many different ways to get into the neurodegenerative conditions and Alzheimer's. It's not probably there's not going to be a single paradigm that explains all of these deterioration of health and deterioration of the health of the brain. When we characterize these dynamics, at least in the physics world, when you think about systems, there are systems that become chaotic, and emergent behavior means that everything is chaotic and it's really impossible to trace back and put some order. And there are systems that are not chaotic, and separating out between the two is a big challenge. And what we would like to try and understand, as you said, is the original signal in the protein changes that starts this process. The way we thought of doing it at some point, and we dropped this approach, was originally you might think you want to have the largest model on Earth that would capture everything, the small molecules, the proteins, the cell, the organ, and combine all of them in one large pre-trained model. And at least my current thinking, and that's, again, my current thinking based on all kinds of analysis we've been doing, is that it's unlikely that we will have a single model that will capture all of that. Similar to what you see in physics, that statistical mechanics captures the statistical behaviors of the, let's say, of a system. Let's say the air in your room now can be characterized by the quantum activity, by the classical activity, by the statistical high-level parameters. So I don't think that there will be one model that captures it all. And the agentic system approach that integrates the different views in a smart way, it's probably the way to resolve that. And that's the direction we are taking. And it enables consuming other efforts and other teams' brightness and methods to tackle different aspects of the question and integrate them together. That's our current thinking.
Thank you. So thank you very much for your interesting presentation. We can see we would have still some few questions, but we need to leave them for later because of the time. So they are still in Slido, will be accessed later. Thank you, saying goodbye to US.
Thank you very much. Bye-bye.
I'll grab this important thing because I have it for a next keynote speaker who this time is coming from Norway. So I'd like to invite here Dr. Ira Haraldsen, who is head of a cognitive health research group at Oslo University Hospital. She, as a clinician and neuroscientist, has worked directly with patients affected by neurodegenerative and cognitive disorders. And her expertise lies in clinical neuroscience and integration of multimodal data. Can I ask Dr. Ira Haraldsen to come here? The stage is yours.
Thank you so much. How is this working? Can I switch by myself? And could you show me the pictures there? Then makes my life easy. It's not very easy to talk after a heavy theory mathematician expert. I am a simple doctor, a real medical doctor, and we are trained to make decisions constantly without theory. We are decision makers in short time frame. This is an ability which builds on something which becomes, by your knowledge, more and more important in the future, as we are very experienced in analog digital twins in our brain. As a doctor, medical doctor, we are very good in fantasizing constantly different factors in multimodal data analogs. I was invited here, and thank you so much, to tell a little bit about our AirMind study, which became in Europe recognized, and I will a little bit confront you with the reality. What are the challenges when it comes to dementia, not only to dementia diagnosis, but maybe to dementia risk diagnostics. AirMind started very early. In 2019, we formed a consortium from 8 European countries. And we wanted to access something which, and I will come back to it, was affordable. We are in a situation that we are, almost our hospitals in Europe or worldwide are on their limitations for a cost explosion which will not be financeable by the recent economic situation. And because we after a while succeeded, I will also go into other projects which we were then able to establish. First of all, there is a complete lack of a biologically defined diagnosis called dementia. That might shock many people, but it is a diagnosis or a risk which is defined by a clinical phenotype, meaning you show behavior and someone has to be suspicious about your behavior. This is not good. we need a rational biological definition which can support your clinical impression of a phenotype. We have in between, during the last 5 years, made success, but also then I will come back to it. The success mainly is due to the biomarkers developed by in industrial big production by Roche Diagnostics. But there is a reason why Roche needed almost 15 years before coming onto the market with finally one biomarker in an industrial relevant manner. And that means that it is not such an easy story, and there are huge reference differences in values between countries, nations, races. And that means, again, that you are on uncertain grounds. So what we needed was to make a timely and accurate diagnosis of something we called mild cognitive impairment. We wanted to make it very simple. We wanted to have a biological idea behind it. And we wanted to make the diagnosis, which takes in Europe between 5 and 8 years on average. Between the countries, we wanted to use 2 weeks. And we wanted, in the best case, in the long run, enable clinically to initiate personalized interventions. And then, very important, we wanted to develop a technology in diagnosing which we could globally distribute. So what is the rationale? Why is this important? We know from empirical population-based studies that 35% of Europe's population will fulfill over 65 criteria for MCI, for mild cognitive impairment. Of these 35%, we expect 14 to 15% fulfilling dementia criteria as the aging effect is increasing. The number of or the percentage of dementia is not increasing in the civilized world, but then the total number is, due to the aging effect, increasing. So, in the beginning, I thought about it. I have a slightly different view on CLARAS. I think CLARAS is essential and in the brain a very good thing. At the moment something is synchronized in our brains, it becomes very difficult. It is only wished if I want to have an intention, I show you my arm, then something is suddenly in the brain coordinated. But mainly when we are in resting state, it should be a lively chaotic in our brains. And in the aging brain and in the brain at risk, this is changing. A hypersynchronicity is then a phenomenon. Okay, what we are doing is we wanted to use old-fashioned EEGs. We wanted to show that with a sheep technology, it is also possible to determine and analyze functional brain networks. And then these functional brain networks we wanted to enrich with genetic information, proteomic information, and then digitalized cognitive testing. And there it was a point that this testing should be non-verbal, not verbally, because we are living in some societies of complexity and societies with highly immigrants, and mainly all kinds of cognitive profiles are built on white Caucasians' abilities to think. and to cognitivize. But that is not good enough. We need to reach all others and all other cultures. So, meaning we wanted to develop with Cambridge Cognition, we wanted then to develop tests where we could be assured that all our citizens could be tested in the same way. Because it is all about standardization of data and data flows and protocols in clinics. What happened then is that the invited clinics in Europe collected all the data in the same manner by using QR codes. And that also included then standardization and calibration of machines. Because you have also a problem if you later develop your algorithms, which kind of hardware have you used? And mainly this will go in as one of the factors why an algorithm is not usable in clinics, depending on which kind or will lower its prediction power because you are using different machines. So this was very important, and we wanted to get access to prospective data in a safe GDPR manner, and meaning that all clinics in Europe then gave directly on an SSD server there by a net-based format their data into the system. This in itself, the data collection was the major point of an innovation, of a service innovation. Because when it comes to algorithms, then you are dependent on not only big data, but also high quality data. And the high quality data would never have been achieved, and we could never have been a reliable algorithm developed if we would have done what we doctors usually do. We are writing on paper and pencil and making notes or on a PC somewhere, and these data are overall and then they have to be transferred. So this was very important to revolutionize. And Eli Lilly is sitting in our advisory board, and they were most happy about the kind we are collected data. They were not so interested in the algorithm in itself, because I always say algorithms are coming and going, data are not coming and going. This is very important. And an algorithm is like fashion. It comes and goes, it comes a new one, and a bright mind is developing the next one. This will always change, but the data will not change. Then another obstacle in AI Mind was to convince people to implement our data collection and also the algorithms. This is not given. People usually like what they have done. And to make a development possible means that we all have constantly to talk to each other and to trust each other. And this is in Europe or worldwide lacking. It's in fact that the neurologists are the most resistant ones. They do not wish to change. And they do not wish, there is also unclarity who owns the patients. Is it a psychiatrist, is it a neurologist, or is it a geriatric internal medical doctor? And they come with different procedures. Again, it is everything about procedure change. I used to say that I am very happy about the political development recently, as it makes it clear, very clear, that we cannot continue as we have done. And I also think that Europe should be grateful that we have a military need at the moment, which helps us understanding that we will need to effectualize our healthcare systems better. And it should be, in the longer run, also of military defense interest, what we can do and how we can manipulate information on our algorithms. Also, this is very important to know, that the risk of AI is high. And if I would be a clever enemy, I would plant information into these algorithms to my own population's advantage. And that needs then to be clear that we have to invest in infrastructure, in cybersecurity, and in quite robust data storages, not only in one tiny hospital, but in nations, and these nations needs to do their work in the same way. Because otherwise, we are in Europe completely lost in 27 nations, if we do everything constantly differently. I told you that we were lucky that we had such an initiative already in 2021. We won then also another call, and this is called eBrain Health. eBrains is one of the 11 infrastructure in Europe under the European Health Data Space. And all kinds of brain information. I heard here the vision of CLARA. It would be important for CLARA to become a part of eBrains, as eBrains is the certified infrastructure in Europe to store brain data. And it is very important that we developed an infrastructure which would, from research to clinic, allow then transfer of knowledge. We opened then as well a third study, which is called FluidX ID, as we also wished to develop together with Roche further biomarkers which may help us predicting earlier. I don't have to tell you this. We will not have already within the next 15 years, not any longer enough money to offer public health. This is a factor everybody knows, and very few people realize what that means, and that will also mean polarization of our societies. At the moment, it is going wrong with our health. There will be unpleasant feelings in many people. So if democracy wants to survive, health is one of the major factors we have to guarantee. Access to health. And this is then the next big step, as we, and I will take the chance, that we are working like all other nations, especially China and the US, on the virtual twin. It is no doubt that the virtual twin is no reality at the moment. No one is able to do so. But we will be soon there that we could build a virtual twin. And it is no doubt that the economy will be there where the virtual twin becomes first a reality. That is also the reason why the US and Europe have decided last year that they want to do this together. And it is also clear that Europe, if we are not shaping up now, and really now, then I think we will be buyers, but we will not any longer be sellers. And to be a seller and a consumer of algorithm, I would highly advise against. Because, again, you will be manipulated for sure. And I cannot understand that not everybody has read the Draghi report very carefully. Because he told us what we had to expect very soon. So why do I think that the twin in Norway, that could be one project together with the US and with Europe, because as you know, Norway is not a member of the European Union. We are one of the most digitalized countries. We have the feeling of trust in our genes. We trust our government. We trust our systems. And that makes us unique. And this trust leads to a willingness to give access to all our data. And we trust that they will not be misused. But it is important that the public is getting a feedback for this trust. And meaning that the economy, if data are our oil, then we need tax systems why Google or Facebook or whomever give back to our society a decent amount of winnings. Therefore, we need economic modeling, how this new modern oil has to be governed by our governments. And therefore, I'm very, very fond of the AI Act. I love it, in fact. And I think the AI Act, and it can come more and more, doesn't matter. We only have to get a pleasant feeling for it and to make it good. We will use, now together with the defense system, we will invest highly into our digital infrastructure, even more than before. And we have, together with the other northern European countries, we have supercomputer systems where we can shut in or we can rent as a project. Tomorrow evening, I want to run one or another algorithm, ergo, we are renting energy and time from the supercomputing, maybe from Finland or from Sweden in addition, so that we make it efficient. You do not need in each country necessarily always a huge amount of energy 24 hours. You switch in the dependency how the developers are making progress. Another way why I don't think we can continue as we do is that the medical data volume doubles now every 70 days. And that is tremendous. So no doctor, in fact, at the moment, we are doing worse than earlier. Our decision making is not any longer as good as it was because we are not able to oversee in each of our specialties everything which has been discovered, and we have too much noise in our brain. This is another very negative aspect. AI could also lead to that we become altogether a little bit more stupid. Because we are constantly bombarded with information, and I lose the focus. This is very important that we are working against. And I think the machines could also maybe help us, or AI agents, as mentioned before, that we would get some help and more free time, computer free time. I guess this will also be a hard thing. I will not go because I see time is a little bit running, but what we are doing now is that we combine data from heart, brain, liver, and endocrine system, and we are also privileged in the 1,000 AI Mind patients that we have all the these data from the same cohort. Then we had synthesized these data to make the variation even bigger, and now we have 100,000 AI Mind participants and hearts and brains and endocrines. We wish that this system will be developed so that others can come in with additional organs, and it will become a click-on model. And all this should be then accessible in the long run for the doctor on a computer to make safer decisions. That have been, at the moment, our use cases. And again, the use case are the organs, but as I mentioned, they are coming from the same 1,000 European citizens. And very important, our platform was established so that EMA and FDA can access it directly and can get access to the split files, meaning the validation cases are of the same description as the development and testing cases. This is very important, and in research mostly overseen, and then you have to establish new clinical studies, and that makes it again very insufficient. Very important was also that this platform holds standards approved by notifying bodies. And that is important as the data control, like in the industry, either in energy industry or wherever, need a 24-hour controlled monitoring. And it was mentioned before that the industry became interested pretty quickly. So Roche is using this data platform for development and testing of and validation of their algorithms, as they, of course, like all others, also wish to have in the future digital biomarkers, not only classical biomarkers. It is also no doubt that we are together with Philips developing our concepts in mobile equipment. I will come quickly back to it. Bosch is has 50% of the revenues of Bosch are coming from the car industry. And as we in Europe do not believe any longer in cars, at least not in classic production of cars, and most of the cars are coming from China, and there will be no way back, we think. Therefore, Bosch has also to rethink where they can make money, as they are one of the biggest companies worldwide from Europe, and they are switching carefully into the sensor fields of medicine. And we are developing now sensors which not only can measure these new blood biomarkers, but also all other kinds of information towards the heart. And today, most of the watches are consumer-based oriented and not good enough for medical prediction. There is too much noise and mistake in the sensors, so the sensors have to make a tremendous jump towards becoming a medical device. And also that is important. And then, of course, the rest of the biopharmaceutical industry is highly interested in our data bank. This is how we envision this. We want to reach patients. We want not to compete against high-tech medical devices or medical procedures. We want to lower the pressure on the hospitals by reaching patients and offering our concepts how we meet patients there where they are. And then those identified at high risk should then be earlier referred to high medical technology. We were lucky that we could align ourselves in our with the UK Dementia data bank. The UK has already adapted their protocols to this kind of, should we call it, screening program, so that we, in the longer run, get access to very diverse population-based information. And it is no doubt that we, especially with the African continent, need to align ourselves, as we are in a situation that there are very different predictors in Africa. For example, ApoE, those of you who knows what that is, it's a gene who is responsible in the white Caucasian race for quite a lot of heart and brain incidents, but that is not the fact in Africa. So we need to get this in place and need a huge predictive power for the global population, as the aging and the transition and the immigration into our countries will not be lower in the future. I think there will be, with climate change, more pressure onto our northern European countries. And therefore, it is very important that we solve the global health problems everywhere and together. And last but not least, I wanted to underline that I think CLARA is a wonderful initiative and a wonderful project, as we need to act now and we need to agree and make negotiations. It's more important to make small steps but with efficiency instead of being too competitive. And the US and Europe are for sure has to work together if we want to keep on track. Thank you so much.
Well, thank you. So this was Dr. Ira Haraldsen from Norway, Oslo, with her Shaping the Future of Dementia Diagnosis with VHT technology. And now, how about the questions? Before we have one, I need to tell you that it's really great that you are a researcher and doctor, but if you are not, you would be a wonderful politician, because you are really impactful. And when we listen to you, we just feel that we have to do something. Do you agree? Yes. She's really good. And we have one question here.
Yeah. I wonder what you mean by responsible AI use from as a physician point of view. Is it in terms of trust or in terms of capitalizing? What do you mean by that?
I'm sorry, I could talk for many hours because the researchers are completely lacking often realistic evaluation of their work. Something can only be exposed to a human being as a patient if it is validated, and if it is validated in a cost analytical way. I can't come to a hospital and tell, oh, I have a very nice algorithm, I want to try that. This is not possible. You have to show that your diagnostic is really delivering a benefit. And a benefit is always a cost-benefit calculation. So, meaning you have to go, like in pharmaceutical industry, you have to go to the health technology assessment. And if something is approved, then the nations today go into these negotiations to the medicinal agencies, and then they evaluate if something is really, the cost benefit is there for your society. That might be very different, in fact, from society to society, and therefore we need research to align more into this system from the beginning. In medical research, of the pharmaceutical industry, 90% of their developments are never making it to the market finally. This is too expensive. We will not have the money. We need the industry to finance, together with us, the aging effect. Meaning we have to offer them access to our data so that they can, in vitro, test with our data bank, for example, their products. And therefore the virtual twin is completely necessary as soon as possible. We need to test the situations, not only the developments, also the effects outside or not inside the patient. Today it is you test a medication in a patient, then the patient is coming back after 3 weeks and you discuss how was the effect. This is not possible in the future. You have to make it more likely that you give the one drug or the other drug. And you have to test that on a machine and make it more likely. So, and then the trust will come, because if you predict the side effects, for example, better, therefore the dementia case is perfect, as we are thinking we are able soon to be able to predict MCI at risk. I forgot to tell, no, I said in the beginning 50% of the MCI patients will become demented. If we could identify those, if we could test would either the one or the other medication help in these cases, it would be wonderful. So as early as possible, because you need to work longer. It's ridiculous that in France it's discussed that we should have strikes on the road because they should work to 64 or what was it. In Norway, nobody had discussed it, and we are working now to 70 altogether, and soon 72. In 2 years, it will elevate it to 72. So there is no way. We have to stay healthy and to work longer. Otherwise, we can't finance it. Yeah.
Thank you. Do we have another question? There's one more in the second row, but you can keep that.
You spoke about noise of sensors. Do you mean some uncertainty of measurement or some interpretability problem of data?
I would have said it is both, but the problem is that the noisiness of the, for example, you can already increase accuracy by having 2 sensors on a watch, but usually the most, at least the Apple Watch and Sony Watch, they will not give you exact enough reliable data about your heart. And then we need also an ECG in addition. So we need quickly a good development of sensors which give you a real life, the ground truth, if you will say so. And the sensors at the moment are not delivering that. So it would make your algorithm on their virtual twin again too unreliable. Not in medicine, we do not. We need accuracy on a very high level. We can't make medical decision on too many false positive or false negative decision making. This is too risky.
Thank you. I think, and again, because of time, this will be all for questions for Dr. Ira. Oh, no, not last one. I'm sorry, I overlooked you.
I have 2 questions.
Even 2.
One is scientific, the other one has to do with public policy. I'm surprised your protocol for assessment does not include language as a early marker of change, which is one of the most important outputs of the brain is the use of language.
Yes.
From evolutionary point of view, it's the last one comes out, and it's the first one to show any sign of deterioration, way before any of the other markers. As part of the CLARA project, I would like to encourage you to think about incorporating language in the pre-MCI, MCI stages. So that's one idea I like to put. The other question I have is about the policy. Again, it relates to CLARA. I think we should cast the problem for Europe as we did in the United States. The problem of dementia is just a proxy for the larger problem of disability that affects cognition, mood, motor function. So I think if we frame the problem in terms of disability and trying to reduce disability, it's more likely to get the attention of the policymakers because the problem becomes far more pervasive. I think your data shows that. You identified some of the major health issues that lead to disability. Dementia being one, but cardiovascular, they all have one thing in common, cognitive changes or mood changes, depression, and so on. Yes. So if we focus on the disability rather than the diagnostic categorization, yes, the public health issue becomes much larger, and the aim would be for CLARA and for your efforts to reduce the duration of disability. The reason
Yes.
we're facing these problems is longevity is beginning to extend the duration of disability. That's what costs public health.
Yeah,
So
I totally agree to both arguments. 40% of the arrhythmia polyclinic patients, also heart arrhythmic, are showing over 65 are showing cognitive decline. 53% of orthopedic hip surgery patients over 65 are cognitively declined, but never correctly diagnosed. So meaning there our algorithm would be an advantage, and there we will send first our bus to those patients, just because you are right. The verbal story is complex, as I guess you are not necessarily referring only to verbal ability, you are maybe also to psychomotoric tempo referring to, and again, we wanted to be assured that we can reach the African market as soon as possible. So that was a clear decision.
Is that complete? Thank you so much. So that was and now time for our third keynote speaker, Heterogeneity in Neurodegeneration, Imaging and Diagnostics. That's topic of Dr. Adam Schwarz, whom am I welcoming to come here. Mr. Schwarz, Dr. Schwarz is Global Head of Imaging Digital at GE Healthcare USA. He focuses on research in medical imaging and neurology with application to drug development. Is that correct?
Yes, that's a good summary.
Great. So
almost
it's
as if
all
I wrote
yours.
it myself. Thank you. Thank you very much, and thanks for the invitation to be here. It's great to have the opportunity. A little bit about myself. I currently work for GE Healthcare. I've only been there less than 2 years. You may know them from the scanners they make, the MRI scanners, the PET cameras, and so forth. That's the kind of the biggest part of the company. But I actually work for a, if this is the laser pointer, maybe not. I work for a part of the company that's called Pharmaceutical Diagnostics. So we're like a small drug company within the larger GE Healthcare, and we develop imaging agents that are used in clinical practice for diagnosis. These could be PET tracers, contrast agents for ultrasound or MRI or CT. We do have two agents that are used in neurology for diagnosis, but I won't be talking about those today. I'll be talking really just in general about some aspects of what I think is some aspects of complexity in neurodegenerative disorders and heterogeneity. And by heterogeneity, I mean differences from one individual to the next individual to the next individual. And I think hopefully it's some food for thought for the CLARA Consortium with its emphasis on high-end computing, but I think at minimum it's something that we should all be aware of when we think about neurodegeneration, when we think about dementia. I'll be talking a fair bit about Alzheimer's disease as kind of the, an example neurodegenerative condition. But as we'll see, hopefully it'll become clear that a lot of these considerations actually generalize. So I'll talk, this talk is really in 3 parts. The first part, I'll talk about some of the things that I mean when I talk about heterogeneity. And then I'll move on to talk a bit about how we can actually measure that in living people. And that's really been a revolution in the last 20 years or so. The advent of biomarkers, be they imaging or fluid-based, which now means we can interrogate the biology while people are still alive. We don't have to wait until they die and go on to post-mortem. And then finally, some of the implications of this in terms of the fact that it does actually matter in terms of people's condition. So we start with Alzheimer's disease as an example, and neuropathologically, Alzheimer's disease is defined by the presence of two types of pathology in the brain. This is amyloid plaques and tau tangles. So these are two types of protein that are naturally present in the brain, but in Alzheimer's disease, they cluster together into these small inclusions, and then on basically a microscopic post-mortem examination of brain slices, you can see these two types of pathology. Okay, so this is Alzheimer's disease, and 30 years ago, you weren't diagnosed clinically with Alzheimer's disease. It would have been dementia of the Alzheimer's type because it had a presentation that was consistent with certain clinical definitions, but it was only after death that you could confirm whether or not it was Alzheimer's pathology driving those clinical symptoms. And so when we think about tau pathology as one of those two, I mean, one thing that came out of many years of neuropathology research was that the pathology seemed to have a sort of a stereotypical spread within the brain, starting in the medial temporal lobe and then gradually spreading through the wider neocortex, and this seemed to be associated with worse symptoms. So there was these staging schemes. This is showing Braak's staging here, which is the most famous. There are other staging schemes, but essentially they all capture this, what was a presumed spread from the medial temporal lobe, hippocampus, into the wider set of the wider parts of the brain. So, okay, so that all seems quite clear. You have amyloid tau, the tau progresses, and you get Alzheimer's disease. But actually, sort of the first layer, if you like, of heterogeneity is just in the clinical presentation. When we think about Alzheimer's disease, most of us would probably think about memory problems and the sort of classic amnestic phenotype. And that in the top left here, that is actually the most common clinical presentation, but it's not the only one. There are other subtypes, the dark red on the left there and the top right, other variants of AD. So even just clinically, there's different ways in which the disease can present. And then there are other types of dementia as well. And then if you try and link those then to more specific, precise cognitive tests and cognitive domains, you can see that they all involve different sets, combinations of these cognitive deficits in a very complex way. I mean, the reason I chose this picture was because these arrows were all crisscrossing all over the place. So there's a lot of complexity here, even just in the way these diseases present clinically. And so, again, it's heterogeneous from individual to individual, and we try and sort of group them into syndromes and so forth, but it can be a very individualized problem. And if we think about, coming back to tau pathology now, if we think about tau pathology more broadly, yes, it's one of the defining pathologies of Alzheimer's disease, but it's also underlying a number of other conditions, other tauopathies, so-called, because they're all defined pathologically by the presence of these tau clumps of these tau protein in the brain. And you can see here in terms of pathological diagnosis and clinical syndrome, there's a lot of complexity here as well. There are lots of different types of presentations. Just because you have quote-unquote tau pathology in your brain, you could have any one of these syndromes. And so why is that? These are all tauopathies, but there is sort of, this is coming into more layers of complexity here now. One potential sort of degree of freedom, if you like, is where in the brain the pathology is found. Because location matters in neuroscience. So you can see this sort of going from left to right on this pathology panel here. These are different brain regions that could be affected by these. Another degree of freedom is the shape or the appearance of these tau clusters in the brain. We have examples here: coiled bodies, neurofibrillary tangles, tufted astrocytes, and you can see these all look differently. So in Ernest Rutherford's terminology, this is kind of stamp collecting. We're just looking at these on slides and saying, hey, they look different, and we give them descriptive names. But it's something that is different in these different brains and associated differentially with these different clinical conditions. Yet another degree of freedom or layer of complexity is the composition. When you do a chemical analysis of these clusters, the actual sort of lengths of the tau proteins can be different, and sometimes in different combinations in these different diseases and different pieces of pathology. So there's a lot of complexity there, and yet another layer of complexity. And this is all just tau, which is just one of the proteins that can build up in dementia conditions, is that there's kind of a lifespan. Basically, these tangles, they start and doing the damage in the brain, and they gradually kill the neuron, and you're left with this ghost tangle, so-called, after the neuron has dissolved around it. And why is this important? It's partly important because this kind of is relevant to what we can detect. What this diagram purports to show here is that different types of stain, different dyes that you use on brain slices to, even neuropathologically, which is as close as we've got probably to the ground truth at the moment, they're differentially sensitive to different parts of this lifespan. If we think about imaging agents, which we'll come on to later, or other biomarkers, we also need to be cognizant of exactly what type and what stage of tau pathology we're picking up. Another example that I think nicely illustrates heterogeneity in both sort of clinical and the pathological sense is frontotemporal dementia. So this is another category of dementia. And so FTD is kind of like the umbrella diagnostic category. But in fact, as we saw that, you may remember on the earlier slide as well, there are different clinical presentations of frontotemporal dementia. You could have behavioral variant, so-called semantic variant, non-fluent variant, and some sort of more motor-related syndromes as well. So these white rectangles here are the different clinical subtypes of FTD. But what are these nice rainbow colors? They are capturing what is the pathology that's actually underlying these conditions. And so you have some cases of FTD that are driven by tau pathology, which we've been seeing earlier in the talk. Others may be driven by a different proteinopathy, TDP43. And there are others as well, other pathologies that underlie frontotemporal dementia. But the reason these look like rainbows up here is because just because you've got, say, TDP43 pathology in your brain, it doesn't mean that you're going to have, say, behavioral variant FTD. You could have that, you could have any one of them. You can see there the colors are all basically all over the map. And this makes it very difficult because in the absence of a biomarker to get at this stuff, if you're trying to develop treatments, which I've spent most of my career doing, or if you have a treatment that's on the market, it's very difficult. If you've got something that's tackling tau pathology, for example, it's a coin toss if someone comes in, whether their disease is driven by tau pathology or not. It makes it difficult to do trials, makes it difficult to treat patients. But it gets even worse because it turns out that not even in dementia, but just in old age generally, the brain is heterogeneous in combinations of pathologies. So these mixed pathologies, the notion that you don't just have one or another of these if you're unlucky in your brain, there are all sorts of pathologies that are found in old age and associated with various dementias. Vascular pathologies, we've heard about amyloid and tau, we've heard about TDP43. Lewy bodies is another one, alpha-synuclein proteins. And this is showing data from a large neuropathology study showing that there's basically, this first graph here is really all the different sort of combinations of pathologies. And you can see there's no real rhyme or reason there. There's all combinations are found. And critically, again, this wasn't even necessarily a dementia population. This was just an epidemiological study. Only 6% of this population on death had no neuropathologies at all, and nearly 80% had more than one. And you can see the histogram there of the combinations of neuropathology. So you can have these layers of different pathologies present at different levels in the same brain. And this just shows a sort of a similar view on the same thing, showing also that a number of these pathologies are strongly age-dependent as well. So that it's to Ira's point, I mean, there's an age effect here that we need to be cognizant of. So just to sort of summarize this first part of the talk, when we think about heterogeneity and we're thinking about dementia or neurodegenerative disorders, we need to be cognizant of the fact that although there may be different kind of clinical labels or diagnoses we make, the reality is that there's a, if this animation is going to work. No, it didn't work. Anyway, the animation was showing those all overlapping into a spectrum. And I think we really need to be thinking of aging, but certainly dementia, as more of a spectrum where it's far more common than not that there's going to be some combination of stuff happening with complex relationships. And so when we think about heterogeneity, there's heterogeneity in clinical presentation, there's heterogeneity in biological presentation, even within what would seem to be a given pathological class, there's lots of heterogeneity there. Things don't always match up in a simple way. And then there's this multiple pathologies, co-pathologies aspect, which is just, there's a lot of layers of complexity here. There's a lot going on, and it's really, really complicated. So what can we do about it? Up until 20 odd years ago, we couldn't do much. Maybe an MRI scan. I mean, MRI is probably the OG in Alzheimer's and dementia biomarkers, but that's not specific for any molecular process. But there's been a real revolution in the last 20 years, and now we're kind of facing, at least in research, like a tsunami of biomarkers for anything and everything. And so this is revolutionizing what we can do in terms of studying living people in this field. So when I talk about biomarkers, the two sort of main types are imaging biomarkers and fluid biomarkers. Now, imaging biomarkers, that's your MRI scans, your PET scans. The big advantage of them is that they give you a 3D map of the brain, of whatever it is you're measuring, so you've got the spatial localization. So that's the real strength of imaging. Fluid biomarkers, on the other hand, don't do that. But there you're measuring some chemical, which is a big plus. But the other, for me, really important strength of fluid biomarkers, be it blood test or a CSF draw, is that you can multiplex. When you take a draw, you can divide that into a number of aliquots. And so from the patient's perspective, from a single procedure, you get maybe 5 different biomarkers out that could provide complementary pieces of information. So which is huge as we're thinking about, again, these layers of complexity. To be able to get all that in a single procedure is massive. And so if we go back to the core AD pathology, and this is where I think that the field really kind of started with this, the biomarker revolution. There are now imaging and fluid biomarkers for amyloid and tau, which, as we've heard, are the definitive biomarkers for Alzheimer's disease. So we don't have to wait to post-mortem anymore. We can do this while people are still alive. We can track them longitudinally. And there are various ways of assessing neurodegeneration, because at the end of the day, it's the loss of neurons or the ill health, shall we say, of brain tissue that is actually underlying the clinical symptoms. One thing we must always bear in mind with biomarkers, though, and it may sound like a silly or reductive thing to say, is that a biomarker measures what it measures. And it's really important to remember the nuance of that. And the amyloid example there perhaps illustrates that quite nicely. So we have a CSF measure of A-beta 1 to 42, which is it's a length of A-beta protein. And then we have the amyloid PET, which is on the y-axis there, the intensity of the PET scan. So they're both measuring amyloid, but they're measuring different aspects of amyloid. So the CSF measure is measuring the free protein, this particular protein in the cerebrospinal fluid. And so that goes down. So the bottom right of that graph is normality, and then you kind of go around the banana as it becomes abnormal. And the reason why it's nonlinear is because you're measuring different things. The accumulated amyloid in the brain is what the PET scan is measuring as it sequesters this protein. But meanwhile, when it's still below the level of detection of the imaging, you can see it reducing in the CSF. So one of the advantages of the imaging biomarkers is they do allow us to sort of maybe have a fairly direct, in some way, certainly in terms of the spatial localization aspect of neuropathology. We can do in vivo neuropathology now with some of these PET tools. And this was some work I did about 10 years ago as these tau PET traces started becoming available in the clinic for human use, and basically translated this Braque staging scheme into living human beings. And it wasn't a given that this would work. The Braque staging scheme is based on certain key brain regions, which you can see illustrated there, but it turned out that it fit the data very well, which was important kind of as a face validity for the tracer early on, but importantly also it gives us a tool now. While people are still alive, we can stage their disease biologically, not just in terms of clinical symptoms. And so this is sort of now giving rise to frameworks that we can be systematic about thinking about diseases in biological terms as well as clinical terms. The so-called ATN research framework for Alzheimer's disease, for example, based on whether an individual is positive or negative for an amyloid or a tau biomarker, tells you, for example, if they would probably be on the Alzheimer's pathological cascade or whether there's something else going on in the brain that might be causing one of these mixed pathologies, for example. And then we have these nice Jack curves where so it's a conceptual tool in a way where you can actually lay these out as a function of time as well. We now know that amyloid pathology can predate symptoms by 10, 15 years, whereas tau pathology is much more closely linked in time to neurodegeneration and clinical symptoms. And so you can see this kind of temporal spacing captured here. And now, as a computational exercise, based on biomarker measurements on individuals, you could make individual curves like this and show where somebody is in this framework. So it can not only be a conceptual tool, it can also be a practical tool that could be applied to synthesize the biological state of an individual. But also we can sort of play with these curves to illustrate other aspects, like we talked about co-pathologies early on. Now, if somebody has additional insults to their brain beyond amyloid and tau, then it's quite likely that the neurodegeneration and clinical aspects of the disease might be accelerated relative to where you might expect based on their tau pathology, for example. They could have lots of Lewy bodies that are driving their clinical decline. And so again, you could sort of imagine how a scheme like this, you can sort of almost infer co-pathologies where we don't have biomarkers for them. Another important aspect when we're thinking about modeling neurodegeneration is cognitive reserve. So this is again, it's a sort of a term for something we don't fully understand. It's a bit like dark matter in astronomy, but it's basically some individuals, for whatever reason, have some resilience to a level of pathology that might be causing a certain level of symptoms in, let's say, the average person. These other people are doing better, relatively speaking. So there's lots of sort of variability and complexity that we can now start to get at in living individuals. And this table is actually well out of date now, but it sort of shows at least 5, 6 years ago a snapshot of some of the biomarkers that are now available. Honestly, these days, it's a real tsunami. There's just a huge number of PET traces being developed, huge number of, especially the last few years, blood tests coming along, different types of proteins. And again, we can see this conceptual tool here for these Jack curves can be used to illustrate and map out all sorts of different aspects of the biomarkers and the disease. And one I just wanted to point out here, which I'm not going to talk about in any more detail, is the notion of neuroinflammation or the immune system and its interaction with the brain and brain health. That's a huge topic of interest at the moment in terms of dementia research generally, but also for the development of new treatments. It's probably the next wave in AD that's coming after amyloid and tau in terms of what's currently in early clinical development. But one of the complexities there is that some of these markers that reflect different aspects of that part of the biology don't have the same monotonic relationship with time, so they could go up and down at different stages of the disease. So it's another layer of complexity because it's hard. Let's say you measure a certain level of that biomarker shown in grey there, which I think is TREM2. you don't know if you're on the downslope of the curve or the upslope of the curve, just on that piece of information alone. But it also actually relates to what we were talking about this morning in terms of maintaining at the health of a broader organism. The brain is not an island. It's not existing in isolation. So this is another important aspect. So to summarize the imaging and biomarkers piece, as I mentioned, I think this has been a real revolution. I think those of us who've lived through this in the last 20 years have sort of seen this in real time, that now we can get at this fundamental pathology in living individuals, at least some of it, because there are still some blind spots. TDP43 is a big one, alpha-synuclein. We're starting, there's one or two kind of things coming out, but they're still, I think, far from what we would ideally like. There is nuances. We always must remember exactly what it is we're measuring, because that can be important. For example, with amyloid biomarkers, in the recent advent of anti-amyloid therapies, these are antibodies that basically suck the amyloid plaques out of the brain. You can see that on PET scans. Now, if somebody comes into the clinic and you want to know if they're amyloid positive or amyloid negative, I'm an imaging guy, but honestly, these blood tests will probably do just as good a job at telling you that. But if you want to see, is the drug having the effect and has it removed the amyloid from the brain, then there's a decoupling there. They don't tell you that. You still need the PET scans for that. So we always must remember these nuances and also the context of use. What is the clinical context in which these are being used? Anyway, this is a huge aspect of current research. So what? There's a lot going on there. I just wanted to sort of close with a couple of thoughts about, so hammer home, hopefully, the implications of this. And one important point about these co-pathologies, as we saw earlier, it's by far the most common situation that the aging brain, let alone somebody presenting with cognitive impairment, will have more than one different type of pathology present in the brain. Now, some of these pathologies, silent bystanders, they're just there, but they're not actually driving any symptoms. Unfortunately, it looks like they do. This is from that ROSMAP neuropathology study that I showed earlier, again, where they took the post-mortem information and then they worked back statistically with the information, the tests they had on the people before they died. And indeed, all these different types of pathology all can contribute in a variable way from individual to the cognitive decline. So these are not silent bystanders, they do matter. And now, as I mentioned, we're starting to get biomarkers for more and more pieces of the pathology puzzle. Alpha-synuclein is a recent one. It's far from a perfect assay, but this just sort of illustrates some recent work showing that measuring this in living individuals, again, you can see that it is associated with a higher degree of cognitive impairment. So just finally, I'd like to just sort of illustrate with a cartoon yet another aspect of why I think this matters. And I've kind of framed this in the context of clinical trials, but I think this also matters for clinical treatment as well as treatments become available. So if we imagine that we have a drug or a candidate drug that is targeting some aspect of amyloid or tau, some AD pathology, and let's just say we think it's going to slow the excessive aging due to dementia, the excessive cognitive decline due to dementia by about 50%. So that's our hypothesis. So if we had a trial population that consisted only of people that only had AD pathology, then maybe a well-designed trial would pick that up, assuming that the drug works as we think it does. But as we've seen, this is actually extremely rare. It's far more likely that you will have participants with co-pathologies, and also the amount of co-pathologies that they have, which ones they have, and how much they're contributing to this cognitive decline is variable from individual to individual. So what all of this means, my arrow's got a bit misformatted there, but what all of this means is that from a clinical trial perspective, it adds statistical noise. It sort of reduces the overall, the ceiling you can have in terms of your therapeutic effect, and it adds variability from individual to individual. And even when we think about going into clinical practice, if someone presents, they may be positive for a certain biomarker, but if that biomarker is contributing only a small amount of their symptoms, the risk-benefit might not be there for them to have, say, a treatment that might have a lot of side effects. So just my last slide now is on some work I did recently to try and sort of get at these different co-pathologies, because as I mentioned, we don't have biomarkers yet for many of them. And so what we did here was we trained a deep learning model using clinical information, but mainly got most of its power from structural MRI scans. And so the notion was, using pathology data to train the model, we could basically determine like the structural MRI fingerprints or the atrophy fingerprint of each of these individual pathologies, and use this to infer the presence or absence of these co-pathologies in the brain. And that's what's shown on the right there. And then when you do that, you can work out how much sort of gain you're starting to get at this problem of co-pathologies in clinical trials and increasing your statistical power. So either you can detect a bigger effect or you can make a smaller trial basically to get the same result. And interestingly, I was at a meeting last week where I learned that this same model, my collaborator Doigu Tosin at UCSF has been working with one of the large pharmaceutical companies to sort of test this in actual phase 3 trial data, and found that it did actually translate pretty well, which is always nice when things generalize. So, summary, there's a lot going on in neurodegeneration, pathologically speaking. There's all sorts of complexity, not only in terms of the upstream drivers, but how much of it there is, where it is, what it's made of, and then how that translates down into the eventual clinical symptoms. But you know, the glass half full piece of that is that the recent advent of all these biomarkers is really providing, you know, as I say, I think it's been a real revolution in the field. And I think these going forward will hopefully be key enablers for personalized medicine. So I'll just close with this this slide here, that, you know, when you kind of think about AD in a sort of textbook, it's often presented in that simplified manner at the top there. But the reality is actually a lot more complex. And I promised last night to make an analogy to the architecture of Prague. Beautiful city, wonderful buildings. You know, there are a few that you could say they're all sort of Gothic style architecture. There are a lot of others that look, you know, like sort of Vienna style, Habsburg style, or whatever it might be, but no two of them are the same. And there's a lot of difference there. When you just walk around Prague, you see all those buildings. Some may look similar to others, some may look more different, but they're all individuals, and it's the same. It's the same with brains, it's the same with people, same with human beings. And hopefully that's something we can start to grapple with in CLARA. Thank you.
We thank you. Another really articulate, colorful and important puzzle. And again, we have already question in the... No, it's yours.
You left out the big elephant in the room out of the picture, which is loss of synaptic connectivity. Amongst the list of pathologies you showed, the most significant one is massive loss of synaptic connectivity, synaptic dysfunction, which is highly correlated, much better correlated with the clinical features, cognitive decline, than the amyloid or the tau, because you have many cases where you have the classic pathologies but absent the clinical features and vice versa. So the relationship between these lesions and the clinical features are not linear or consistent, although with some subsets it's true. That's one point. The other point is the first paper on diagnosis of Alzheimer's was 1985, which called for, it was in the Archives of Neurology, which established the criteria for neuropathological. But in addition, it called for multimodal diagnosis, amongst them being the notion of biomarkers. and it called for the development of ligands, which later on led to the Pittsburgh compound. But one thing that hasn't happened is the development of ligands for synaptic dysfunction. And that's what I think the challenge is for you and for GE, to develop those ligands for the biomarkers associated with mitochondrial dysfunction, synaptic loss, and so on. I'd like to hear your thoughts. I think that would be a great opportunity for GE and CLARA to collaborate in developing those, because I think it can be more fantastically significant.
Yeah, yeah. No,
by the way, I enjoyed your talk. Just
thank you.
Not being critical.
Yeah, I agree with you about the neurodegeneration aspect and the synaptic dysfunction. There are different ways of getting at that. I used to work a lot in functional connectivity, and then I've done studies or been involved in studies looking at things like recent ligands getting at mitochondrial, well, dysfunction maybe in inverted commas, but the mitochondrial complex 1. So I have a paper with the guys in London on, we looked at mitochondrial complex 1 tracer, another PET tracer looking at sigma 1, I think, and then maybe SV2A might have been in that study as well. And I also, when I was at Lilly, I did a collaboration with Yale when the synaptic density, the SV2A marker first came out. And I think one of the challenges, because I was sort of coming at this from a sort of drug development perspective, but one of the challenges is that these markers have tended to have fairly small effect sizes. If you look at the difference between, for example, the MC1, the mitochondrial one, there was a slight difference between Alzheimer's and the healthy controls, but it wasn't huge. And I think that's part of the challenge as well, is sort of how, in drug development, how would you use them instead of something that's more closely related upstream with the target of your therapeutic, which that's the way people think in the pharma industry, or some measurements of clinical outcome or clinical tests, because that's standard of practice in medicine, and that's what the regulators tend to want to see. So there's always been a bit of a valley of death there between getting at biomarkers of the synaptic dysfunction or other aspects of neurodegeneration. I think fundamentally it's what, when you're developing a drug, what could it be used for in terms of making a decision? Is it going to help get the drug approved? Is it going to help find the dose? Is it going to help demonstrate that we think the drug's working in a small population and we can justify the investment into a larger trial? Is it going to help convince the FDA or others that there's a biological effect here, and if we saw something on a statistical sense, it wasn't just a fluke, but there's some biological evidence lining up behind it through the biomarkers. So those are some of the things that people think about in drug development. But I think fundamentally there hasn't really been up to now, a compelling case of when to use these. I think everyone's interested, but they're sort of waiting to be convinced. And the SV2A, I think everybody expected that there'd be bigger effect sizes, bigger differences between disease and healthies, and it just seemed quite subtle. And maybe that's what it is biologically for whatever reason, and those subtle effects are enough to have the clinical impact, but they're difficult to use as a biomarker in terms of your dynamic range.
Tom Budinger, a number of years ago, had developed imaging calcium ion distribution. I don't know what's happened to Tom Budinger, but that's one. The other one is Rodolfo Linus had developed calcium imaging in vivo rather. It would be nice to be able to mimic those for GE, to be able to do nano imaging of various circuits in the brain. That would be phenomenal, because now that would allow us to analyze circuit dysfunctions.
Yeah, yeah. But also, as you hinted at, I think there's a lot of nonlinearity, complexity in that aspect. And, you know, there's a necessary aspect of simplification to some degree in the clinic, but I can speak to that. But certainly when you're doing drug development, you have to boil things down to something that's relatively simple and robust, which I think when things get complicated, even just that non-monotonicity, then suddenly it's like, oh my God, how do we know we're using this correctly? So there's plenty of scope for more in biomarkers for sure, and maybe you're right in that the sort of the neurodegeneration aspect, including the function, is still something that hasn't been fully tapped.
Thank you. I like the deep questions we are getting from this part of podium. I would like to remind that if anybody else is just shy to ask question and is here, also yourself can use Slido, and we will find it there and answer now or later. And thank you for Dr. Schwarz for really nice presentation.